All right, so get this. We're diving deep into something called autophagy. Okay. You sent over this article. Right? It's from Assay Genie.
Right.
And it's titled, Understanding the Autophagy Pathway, a critical process in cellular maintenance. And the more I read it, the more I was like,
okay,
how have I not heard about this?
Right.
It's like your cells are doing this intense cleanup and recycling operation. And it affects like everything from how you age to how your body fights off infections.
It really is fundamental. And it's fascinating how it's like essential for survival, but also can go rogue and contribute to disease.
That's what I thought was so interesting.
Yeah.
So before we get into all that, let's start with the basics.
Okay.
What is autophagy?
Autophagy means self-eating.
Oh, wow.
Which might sound a little gruesome.
Yeah.
But it's actually a very sophisticated system.
Okay.
Essentially.
Yeah.
Your cells have this way of identifying their own damaged components.
Okay.
Things like misfolded proteins or worn out organelles or even invading pathogens.
Oh, wow.
And
so they can tell what's bad.
Yeah, they break them down into these reusable building blocks.
So it's like they have their own waste management system.
Yeah.
But it's way more advanced than just like throwing stuff out.
Yeah, they're sorting through everything, breaking it down.
Wow.
Reusing the good parts.
That's incredible.
And this isn't just happening randomly.
Okay.
There are specific pathways and signals that control autophagy.
Right.
Making sure it happens at the right time and in the right way.
Okay, so
it's like a very tightly regulated dance.
Okay.
Of molecules and cellular structures.
So it's not just one method.
You're right to ask that. Okay.
Because there are actually 3 main types of autophagy.
Oh, wow.
Macroautophagy.
Okay.
Microautophagy.
Right.
And chaperone-mediated autophagy.
Right.
And each uses a slightly different approach to achieve the same goal of cellular cleanup.
So macroautophagy.
Yes.
That's the one I've seen.
Okay.
Described as involving these little Pac-Man-like structures.
That's a great visual.
Okay.
Because it captures the essence of macroautophagy.
Okay.
The Pac-Man in this case is called an autophagosome.
Autophagosome.
And it's a double-membraned vesicle that forms within the cell.
All right.
And its job is to engulf the cellular material that needs to be recycled.
So the autophagosome is like a tiny garbage bag.
It is like a tiny garbage bag.
Going around and collecting the damaged goods.
Yes.
Okay, cool.
So then
what happens next?
This is where things get really cool.
The autophagosome, now filled with cellular debris, fuses with another cellular compartment called a lysosome.
Lysosome.
And you can think of the lysosome as the cell's digestive system.
Oh, so
it's packed with enzymes.
It's like sending the garbage bag to the incinerator.
Exactly.
Okay.
Everything gets broken down into its basic components.
Awesome.
And those basic components, things like amino acids, sugars, and fatty acids, they're then released back into the cell.
Oh, wow.
To be used as building blocks for new molecules.
So it's like the ultimate recycling program.
It is.
That's so cool.
Yeah.
But you mentioned there are other types of autophagy too.
Right.
What's different about microautophagy?
Microautophagy is a bit more direct.
Okay.
Instead of forming a separate autophagosome, the lysosome itself directly engulfs the material that needs to be broken down.
So no middleman.
No middleman.
Straight to the digestive system.
Straight to the digestive system.
Okay, cool. What about chaperone-mediated autophagy?
Chaperone-mediated autophagy.
That one sounds
more complicated.
A bit more selective.
Okay.
In chaperone-mediated autophagy, CMA, specific proteins are targeted for degradation.
Right.
These proteins have a special signal sequence.
Okay.
That's recognized by chaperone proteins.
Chaperones like at a school dance.
That's a fun way to think about it.
Okay. To make sure the kids don't get into too much trouble.
That's right.
Yeah.
These chaperone proteins bind to the target proteins and escort them to the lysosome.
Okay.
But here's the catch.
All right.
The lysosome membrane has a special receptor that only recognizes proteins bound to these chaperones.
Oh, so it's like a very exclusive.
It is. Only proteins with the right invitation get access to the lysosomal breakdown party.
I like that.
So this level of selectivity allows the cell to fine-tune its protein recycling.
Targeting specific proteins without disrupting others.
It's amazing how intricate and organized
it is.
This whole process is.
It's amazing.
But how does the cell decide what needs to be recycled?
That's a great question.
Like, how does it know what's trash and what's treasure?
That gets at the heart of how autophagy is regulated.
Okay.
There are these specific signals and sensors that alert the cell to the presence of damaged or unwanted material.
So what are those signals? How do they trigger this whole process?
One of the most important signals is nutrient deprivation.
Okay.
When the cell's running low on essential nutrients, it triggers this cascade of events that leads to the activation of autophagy.
So it's like the cell is saying, times are tough. We got to recycle some of this old stuff.
Exactly. It's a remarkable survival mechanism.
Wow.
But nutrient deprivation isn't the only trigger. There's other signals like oxidative stress.
Oxidative stress.
Which is damage caused by free radicals.
Oh, those nasty little molecules that can wreak havoc on our cells.
And there are even signals that come from the cell's own internal quality control systems. If a protein is misfolded or an organelle is damaged beyond repair, there are mechanisms in place to tag those components for autophagy.
So there are multiple layers of surveillance.
There are.
Making sure nothing slips through the cracks.
Yes.
But I'm curious about the actual trigger itself.
Like
what flips the switch that tells the cell, okay, time to start this process.
That's where things get even more fascinating. There's this specific protein complex.
Hold that thought.
Okay.
We're going to have to come back to that.
Called the ULK1 complex.
ULK1 complex.
And you can think of it as the master regulator of autophagy.
Okay, so this is like
when the cell...
the general giving the orders to start this whole...
When the cells sense those stress signals we talked about, like nutrient deprivation or damage, it activates this ULK1 complex.
Does it directly start building those autophagosomes?
Not quite. The ULK1 complex actually sets off this cascade of events, activating other proteins and signaling pathways. One of the key players in this cascade is another protein complex.
Another complex.
Called the Beclin-1 VPS34 complex. It really is a beautifully orchestrated process.
It's like uncovering a secret society inside the cell.
It is. And the Beclin-1 VPS34 complex has a crucial role. It helps to generate a specific type of lipid called PI3P.
PI3P.
Which acts as a kind of marker on the cell membrane.
A marker?
It's marking the spot.
What's it marking?
Where the autophagosome will start to form. Think of it like laying the foundation for a building. The PI3P molecules create a platform on the membrane.
And that's where the autophagosome will begin to assemble.
So we got the signal from the ULK1 complex. We've got this marked spot on the membrane. How does the autophagosome actually start to take shape?
This is where those ubiquitin-like conjugation systems come into play. You asked about ubiquitin earlier. And it's a fascinating molecule. Think of it as this tiny tag that the cell attaches to proteins.
Like a sticky note saying, take me out.
Exactly. In the case of protein degradation, ubiquitin tags proteins that need to be broken down by the proteasome, which is another cellular cleanup system. But in autophagy, these ubiquitin-like systems have a different role. They help to build and assemble the autophagosome itself.
So instead of tagging things for destruction, they're tagging the components that will do the destruction.
That's right.
That's really cool.
There are two main ubiquitin-like systems involved in autophagy. The ATG12 system and the ATG8 system. They work together almost like construction crews to link together proteins and lipids, gradually building that double membrane structure of the autophagosome.
So the autophagosome is starting to take shape, but it's still empty.
It is?
How does the cell decide what gets loaded into this cellular garbage bag?
That's the crucial step of cargo selection. And this is where those adapter proteins come into play. You mentioned P62 earlier. And it's one of the key players in this process.
So this is the quality control inspector.
You could say that P62 has this very specific job. It recognizes and binds to proteins that have been tagged for degradation, often by ubiquitin. Remember those ubiquitin tags we talked about? Well, P62 can bind to those tags and act as a bridge, linking the tagged proteins to the growing autophagosome.
So it's like saying,
Okay.
aha, you've got a ubiquitin tag.
Yeah.
You're coming with me. You're coming with me.
And then it drags the tagged protein over to the autophagosome.
That's a great way to visualize it.
And tosses it in.
And there are other adapter proteins besides P62.
Okay.
Each with their own specificities and roles.
Right.
Together, they ensure that the right cargo is loaded into the autophagosome.
It's amazing.
It is.
It's like this incredibly intricate sorting system.
Yeah.
Making sure everything is in the right place.
Right.
At the right time.
At the right time.
But what happens once the autophagosome is loaded up?
Right.
We talked about it fusing with the lysosome. But how does that actually happen?
That's a great question.
Yeah.
And it involves a whole other set of proteins.
Okay.
Think of it like this.
Okay.
The autophagosome and the lysosome are like two spaceships docking in space.
Oh.
They need to align perfectly and then fuse their membranes together.
So what are the pilots guiding this cellular docking maneuver?
There are specific proteins called snare proteins.
Snare proteins.
That play a crucial role in membrane fusion.
Okay.
They act like tethers, pulling the autophagosome and the lysosome together.
Okay.
And then helping to merge their membranes.
So it's not just a random collision.
It's not.
It's very
deliberate.
Deliberate and controlled.
And controlled.
And then once they fuse,
yes,
we have that super compartment
right.
where all the breakdown happens.
Yes.
What was it called again?
An autolysosome.
Autolysosome.
It's the powerhouse of cellular recycling.
Right.
Where the enzymes from the lysosome get to work, breaking down the contents of the autophagosome into their basic building blocks.
Okay, so we followed this journey of cellular waste.
Yes.
From being tagged and sorted to being broken down and recycled.
Yes.
This whole process is incredible.
I know.
But I have to ask.
Okay.
Why is it so important?
Yeah.
What happens if autophagy doesn't work properly?
That's where things get really interesting and a little bit scary.
Oh, no.
Because as crucial as autophagy is for cellular health, it's also a process that can go wrong. And when it does, it can have serious consequences for our health.
So what kind of problems can arise when this cellular cleanup crew goes on strike?
Well, one of the biggest areas of concern is the link between autophagy and diseases like cancer.
Cancer?
Yeah.
But I thought you said autophagy helps to eliminate damaged cells.
That's true.
Which should prevent cancer.
It is one of the reasons why autophagy is considered a tumor suppressor mechanism in the early stages of cancer.
Right.
By clearing out damaged cells, it can help to prevent them from becoming cancerous.
Right. So what changes in the later stages?
It's a bit of a paradox. While autophagy can help to prevent cancer in its early stages, once a tumor has developed, it can actually help cancer cells survive and thrive.
Wait, so it's helping the enemy?
It is.
How does that work?
Cancer cells are notorious for their ability to adapt and survive in these harsh environments. They often face nutrient limitations and other stresses, and they can actually hijack autophagy to overcome these challenges.
So they're using the cell's own recycling system
to
fuel their growth.
To fuel their growth and spread.
That's devious.
It is. And that's why understanding the role of autophagy in cancer is so crucial. Researchers are actively exploring ways to manipulate autophagy as a potential therapeutic strategy.
So you might want to boost it to prevent cancer. And in other cases, you might want to block it.
You might.
Stop tumor growth.
Exactly. It's all about finding the right balance.
That makes sense.
And it's not just cancer. Autophagy is also implicated in a wide range of other diseases, including neurodegenerative disorders like Alzheimer's and Parkinson's.
We're definitely going to need to dive deeper into those connections.
We are.
All right, so we've established that autophagy, which is an incredible process, that's usually a good thing. But it can go haywire.
Yes, it can.
And contribute to some serious health problems. We left off talking about neurodegenerative diseases. Like Alzheimer's and Parkinson's. How does autophagy fit into the picture there?
Well, one of the hallmarks of these diseases is this accumulation of misfolded proteins in the brain.
Like amyloid plaques in Alzheimer's.
Exactly. And what's interesting is that autophagy is one of the main mechanisms the brain uses to clear out these toxic proteins.
So it's like the brain's garbage disposal system.
Yes, you can think of it that way. Constantly working to remove the buildup of cellular debris.
So if autophagy isn't working properly, it's like the garbage disposal gets clogged. And all that waste starts to pile up.
That's a great analogy.
And that leads to problems.
And that's what researchers believe is happening. In many neurodegenerative diseases, there's growing evidence that impaired autophagy contributes to the buildup of those toxic protein aggregates, ultimately leading to neuronal damage and cell death.
This is fascinating.
It is.
But it's also kind of scary. And it makes you wonder if there's anything we can do to boost autophagy, especially as we age, to help protect our brains.
That's one of the big questions researchers are trying to answer right now.
So what are some of the things that they're looking at?
Well, one approach is to focus on lifestyle factors. There's evidence that things like exercise, calorie restriction, and even certain dietary components can stimulate autophagy.
So it's like giving the brain's cleanup crew like a little extra nudge.
A little extra motivation.
To get to work.
Yes.
Are there any specific foods or nutrients?
There is a lot of interest in the role of diet. Some studies have suggested that compounds like resveratrol, found in red grapes, spermidine, found in aged cheese and mushrooms, might have autophagy-enhancing effects.
So a glass of red wine and a cheese plate.
Maybe.
Could be good for the brain.
It's possible.
I'm just kidding, of course.
Of course.
Always talk to your doctor. Before making any dietary changes.
Always consult with a healthcare professional.
But seriously. This connection between diet and autophagy.
It is fascinating.
It's really fascinating.
There's a lot to explore there.
Are there any pharmaceutical approaches?
Absolutely. There are several drugs already on the market that are known to modulate autophagy. Although they weren't originally developed for that purpose.
So they're finding new uses for existing medications?
That's right. Sometimes these serendipitous discoveries happen.
What are some examples?
One example is rapamycin.
Rapamycin.
Which is an immunosuppressant drug that's been shown to induce autophagy. Another is metformin. A commonly used diabetes drug. That also seems to have autophagy modulating effects.
So these drugs originally developed for completely different conditions.
I know, it's amazing.
Might hold the key to treating. Or even preventing neurodegenerative diseases.
Potential's huge.
This whole deep dive has been a real eye-opener.
It has.
I feel like I've gained this whole new appreciation for the complexity of our cells and the incredible processes that are constantly happening inside us.
It's a reminder that even though we can't see it with the naked eye, there's this whole universe of activity going on at the cellular level. And it's ultimately what keeps us alive and functioning.
We've covered a lot of ground today. From the basic definition of autophagy to its role in health and disease. And even some potential therapeutic strategies. It's clear that this is a fascinating and rapidly evolving field of research. With the potential to revolutionize how we approach health and longevity.
I think the key takeaway here is that autophagy is this incredibly powerful process with both beneficial and detrimental effects. The challenge now is to understand how to harness its power for good. To promote health and prevent disease.
Absolutely. Huge thanks to our listener for suggesting this topic.
Thank you.
It's been a truly enlightening deep dive.
It has.
To all our listeners, we encourage you to keep exploring.
Keep learning.
This fascinating field.
There's so much more to learn.
There is so much more to learn.
And new discoveries are being made all the time.
Absolutely. That's it for this episode of The Deep Dive.
Thanks for listening.
Until next time.
Stay curious.
Stay curious and keep those brains hungry for knowledge.